Hi, I'm George Hammond, conference producer at Technology Networks, and I'm here to introduce the final talk of the day: Accelerating Precision Medicine through Precision Dosing, Leveraging PK/PD Modeling for Optimized Therapeutic Strategies. I'm incredibly pleased to welcome Dr. Sharmeen Roy. Passionate about leveraging technology to amplify the impact of the pharmacist, her career spans pediatric clinical pharmacy, clinical research, and pharmacogenomics, with leadership roles at the University of Chicago Medical Center and Pipeline RX, and in her current role as the Chief Strategy and Science Officer at DoseMe. Sharmeen received her Doctor of Pharmacy degree from the University of Illinois at Chicago, is a board-certified pharmacotherapy specialist, and completed a pediatric specialty pharmacotherapy residency at Texas Children's Hospital. A very warm welcome to you, Sharmeen. Following the presentation, we will also have a Q&A session, and we welcome any questions that you may have. Please ask questions using the Q&A system to the right-hand side of the video player. And now, without further ado, I will hand over to Sharmeen to begin her presentation.
Thank you. Good afternoon, everyone. I'm very excited to be talking to you about how we can leverage PK/PD modeling to optimize therapeutic strategies and achieving precision medicine and precision dosing. So, we live in a time where precision medicine is no longer just a concept, but a practical approach that can significantly improve patient outcomes. At DoseMe, we've developed an advanced software, DoseMe RX, that leverages complex pharmacokinetic and pharmacodynamic models to provide precision dosing recommendations right at the bedside to the clinicians. And today, my objective here today is really discuss and delve into the paradox of precision medicine and traditional drug development. We will be talking about the ongoing challenges of drug development, focusing on average doses rather than individualized therapy. I'll be describing this clinical decision support through model-informed precision dosing, or MIPD, and explaining how MIPD can improve treatment outcomes by identifying the optimal dose for each patient. At the end, we will talk about some real-world cases and examples of how we've been successfully able to integrate this into our clinical practice, ultimately improving patient care. Now, precision medicine versus precision dosing, let's get to the basics here. I'm sure many of us, myself included, use these terms interchangeably, along with others like personalized medicine. So just to break it down, precision medicine is the right treatment at the right time to the right person. And precision dosing is a further refinement of that, where we individualize the dose based on unique patient characteristics. So looking at patient data such as genetics, demographic information, some integrating clinical information along with drug response monitoring through pharmacokinetics and pharmacodynamics and therapeutic drug monitoring. So it's sort of like a car with an engine that requires a specific amount of fuel to run efficiently. And if you fill it with too little or too much, you risk stalling or damaging the engine. Precision dosing ensures that each patient gets that exact amount of fuel, or in this case, medication, that they need to achieve optimal health. And by understanding how a patient absorbs, distributes, and metabolizes the drug, we can adjust the dose in real time and achieve the best outcome, minimizing side effects while maximizing benefits. So in short, precision medicine allows us to customize treatments, where precision dosing gives us the tool to fine-tune that customization, ensuring each patient gets exactly what they need, nothing more, nothing less. Now, we get to the crux of the problem here, the issue here. We need to discuss, why is this important? Traditional dosing, as we know it, is a one-size-fits approach, and it faces limitations addressing the interpatient variability, particularly with drugs that have narrow therapeutic windows, such as antimicrobial and cancer therapies. And precision dosing eliminates this variability by tailoring the treatment to our patient. Now, the issue is with our drug development process. This is an abbreviated depiction of the approval pathway, but I want to highlight the paradox of drug development. The problem we are facing is that for a new drug to be approved, it starts out being studied in phase 1, phase 2, and phase 3 with healthy, well-defined subjects. We then get to the FDA approval, and that's when we start seeing its application in real world patient and also through phase 4 activities. After the approval is where we learn about how this impacts real world patients. We know that the patients that we're treating every day are not often represented in the population cohorts that are in the study. And that therein lies the problem. Specifically, we have a population that's aging who are often underrepresented in the clinical study or excluded from these studies. It's interesting to note that although 61% of new cases of cancer occur among the elderly, recent studies indicate that elderly comprise only 25% of participants in cancer clinical trials. There's also a concern with dosing in obesity. And in the US, it's predicted that US obesity rates will approach 50% prevalence by 2035. There's also other groups like neonates, pediatrics, pregnant women that are not well represented in these clinical studies. And so what happens is that once we get to the bedside application, there's a lot of trial and error dosing that's happening and less of precision dosing. And we want to create the tools available to these clinicians to be able to get closer to precision dosing and less of the trial and error method. In those real world patients, this is what the clinical studies are establishing. So clinical trial established evidence of safety and efficacy for standard doses in population, while clinicians are treating individual patients whose response to drug therapy can widely vary. We can utilize technology here and modeling software using MIPD. Includes utilizing advanced PK and PD models to predict how a patient will respond to a drug. These models integrate various patient-specific data to stimulate and optimize dosing regimen. And by using these models, clinicians can make informed decisions about most appropriate doses for each of their patient, enhancing efficacy and minimizing adverse effects. Now, this is not a new concept. The first PK studies published were established in the 1960s, and they established the importance of monitoring of drugs. FDA and EMA have required population PK studies from 1990s onward in new drug applications and approvals and in clinical trial design, and they're starting to enter a common clinical practice now. The FDA and regulatory bodies globally have begun supporting data-driven optimization. And it's interesting to note that the first model-informed precision dosing tool was developed in the late 60s by Shiner, and it was looking at dosing and management of anticoagulation therapy. Now, you're probably wondering how it's going. I'm an optimistic, so changes have been happening in the right direction, but we need to do better. And this timeline will show you some of the changes that have happened from the 90s onward. It wasn't until 1993 that the FDA and Congress required inclusion of women in clinical trials. In early 2000, specifically 2002, the passage of Best Pharmaceuticals of Children's Act included and encouraged drug evaluation in children for drugs meant to be used in children in addition to adults. In 2020, FDA released guidance on enhancing diversity in clinical trial populations that included the inclusion of elderly patient population to better understand how a drug response and drug exposure in that patient population. In 2021, FDA launched Project Optimus through their Oncology Center of Excellence, which was aimed at reforming the dose selection and optimization paradigm in oncology. And just as recently as last year, there was further guidance provided by the FDA in drugs that might have certain adverse effects on kidneys or liver to be tested on people with kidney and liver disease. Now, keeping in mind our end goal, which is improving patient care and really keeping our patients in mind. And what we're trying to do ultimately is improve efficacy, reduce adverse events, and enhance patient outcomes. And we're doing this by increasing patients that get to therapeutic target attainment quicker, decreasing toxic levels, and increasing the proportion of patients that are achieving better outcomes. The role of PK/PD modeling and precision dosing needs to go beyond the clinical trial setting, beyond research and to the bedside. And that's where we want to leverage it at the bedside with the clinicians. The need to study drugs in more diverse patient population has been more and more widely recognized. MIPD can utilize the characteristic differences in drug exposure and responses for underrepresented population. There have been technological and clinical advances that have enabled this translation from research to bedside. With all that said, though, MIPD often is perceived as too complicated to incorporate into clinical practice. However, there is no shortage of evidence in literature that uses technology get closer to precision dosing, and individualizing dosing. Dosing improves outcomes. And I just want to kind of explain how we do that by using DoseMe Rx. We want to use the power of PK/PD modeling at the bedside by simplifying a complex method utilizing Bayesian forecasting software, also known as MIPD, providing that clinical care with limited resources, and also real-time application of this technology. As you can see, it's taking in not only the characteristics of the patient, the doses, pathology, and genomic profile, incorporating that with the algorithms built in, as well as published PK/PD model, and generating doses so we decrease the amount of trial and error that is happening, and starting out with individualized dose calculations for those patients at the bedside. And this is a screenshot of what it looks like of Bayesian dosing in action. This is a drug called vancomycin, showing how we can individualize dosing in patient. You know, we talk about how to improve dosing in drugs that are newly being approved. However, what we're also realizing is that due to the limitation that exists when some of these older agents were approved, we were still trying to optimize dosing for some of those medications, particularly in infectious disease. Vancomycin is one of the single most used antibiotics in hospital settings. It's also well known that it can cause nephrotoxicity. Believe it or not, it was approved in the 50s, and we're still trying to get it right with this medication. In 2020, vancomycin is also a success story, and in 2020, there were several key recommendations that came out of the consensus guideline that was published around dosing methods and targets for vancomycin. Some of the key recommendations here were in adult and pediatric patients with suspected or definitive serious MRSA infection, an individualized target of AUC over MIC ratio of 400 to 600. You can start to begin monitoring with therapy within the first 24 to 48 hours. Utilizing conventional methods as trough-only monitoring with a target of 15 to 20 was no longer recommended based on the efficacy of nephrotoxicity data in patients with serious infection. And the preferred approach was to monitor AUC through the use of Bayesian software programs, which are embedded with PK model based on richly sampled vancomycin data as the Bayesian prior, using 1 or 2 vancomycin concentration with at least 1 trough. And due to the timing of these guidelines, they were published in 2020. The world was dealing with other priorities, so it's taken some time to implement these changes. In 2023, Dr. Sheets, who is an expert in the field, conducted a survey to see what the status was. How were people dosing vancomycin? Of course, he utilized a very reliable tool called X, formerly known as Twitter. And we learned that most people were utilizing and still using conventional methods of dosing by troughs only, which have, as you can see, not recommended by or supported by the guideline. So what we wanted to do was look at real-world sample of data for patients that were dosed using Bayesian software, DoseMe RX. And this data was presented at the most recent ECMID meeting in Barcelona. Dr. Sheets was kind enough to let me borrow some of his slides from our abstract that was presented. So the purpose, the abstract that was presented was big data, Bayesian truths, no vancomycin trough concentration target is sufficiently precise for safety or efficacy. What we wanted to do was interrogate a large cohort of internationally represented real-world patients with Bayesian estimated vancomycin AUC exposure and measured trough concentration, and assess categorical agreement between those two groups. We included adult patients treated with vancomycin in 2022 and with therapeutic drug monitoring performed as part of their routine care. We excluded patients that didn't have plasma concentration, were on hemodialysis, or had less than 1 day of treatment. The data was obtained. Unique patients and dosing events were quantified through concentration or minimum concentration, Cmin. And patient individualized AUC was calculated using Bayesian methods with 4 validated models. And as you can see from the primary analysis of the results, this was a large number of subjects, over 35,000 patients, and analyzed over close to 300,000 courses. In this primary analysis, you'll see in the scatter plot with the vancomycin AUC versus Cmin, and we looked at categorical agreement or disagreement. The red categories are the most worrisome due to major discordance, but low incidence. The low troughs that were at goal AUC, which were close to 30% here, those are concerning because from a clinician point of view, based on these low troughs, you are likely to increase the dose inappropriately with the risk of potential adverse effects. So in summary of that study, we found that only 23% of paired Cmin and AUC were at target, demonstrating the variability of exposure in this patient population. There was categorical disagreement between Cmin and AUC for about 33% of the patients, with the most disagreement being with low Cmin but at target AUC, where clinicians are at risk of overdosing patients based on troughs alone. So these findings further support AUC therapeutic drug monitoring and challenge the Cmin as an adequate vancomycin AUC proxy. And this is important because, although we have a consensus guideline, this was looking at real-world data that was collected through our software to show that it supports the guidelines, and based on how clinicians are utilizing the software, they're able to demonstrate that using the Bayesian forecasting software produces better results and achieves target much more appropriately and precisely. There have been other recent publications to show improved outcomes with real-world application of our software. As you can see, on the top here, Covington studies, that it demonstrate that there was using acute kidney injury incidence with Bayesian dosing software versus 2-level first-order area under the curve based dosing of vancomycin with piperacillin and tazobactam. It showed that there was a reduction in AKI, an increase in attainment of target, and increasing in usable levels. We also had a cancer center in Brazil that demonstrated a 70% decrease in AKI rates, resulting in significant cost savings by switching to vancomycin monitoring from trough to concentration to area under the curve. One of the biggest challenges that clinicians face are that the patients we're treating today are getting more and more complex. MIPD in special populations highlighting the versatility and necessity of MIPD in treating these special patients and challenging the patient groups such as pediatrics, elderly, obese individuals, and patients undergoing dialysis, where traditional dosing strategies often fail to ensure efficacy and safety. Here, we had some evidence, again, showing overall high agreement between 1 versus 2 concentrations in AUC 24 in patients with obesity. And on the top right, showing a pediatric population and demonstrating a decreased risk in toxicity. To further illustrate the impact of MIPD, let's delve into the advancement of beta-lactam dosing. Beta-lactams such as cefepime or meropenem are reserved for critically ill patients and are dependent on maintaining adequate concentrations. TDM is still not the standard of care. However, software can help you take care of those complex cases, allowing you to use pharmacodynamic targets such as % of time above MIC to help guide dosing with or without TDM. While we have seen significant advancements in infectious disease, benefits of MIPD extend far beyond antimicrobial therapy. There is application in other therapeutic areas such as oncology. Busulfan used primarily in conditioning regimen for bone marrow transplantation. Currently, the practice is to obtain upward of 10 to 12 post-infusion concentration. As you can imagine, that is very burdensome, not only on the clinical team that's taking care of the patient, but the patient themselves. Busulfan TDM in these patients, in these bone marrow transplant patients, can be simplified using DoseMe Rx and going from 10 to 12 post-infusion concentrations that would be required to only 2 samples, which in turn, again, reduces the requirement for not only the blood collection samples, lessens the distress on the bone marrow transplant patients, and greatly reduces costs by decreasing the unnecessary assay. This figure here on the side is showing our predicted versus measure. There was a DoseMe Rx analysis that was done comparing predicted versus actual concentration in pediatric patients. This manuscript is in progress. But as you can see, there's good agreement between using the limited sample strategy in these patients. And because these are pediatric patients, you can imagine that the limited sampling strategies were very beneficial in these patients because we try to limit how much blood draw is needed from these smaller patients of ours. Application in other areas such as infliximab, therapeutic drug monitoring and individualized dosing recommendation has been incorporated in the guidelines. It's more cost-effective than empiric dose escalation, improving clinical scores, CRP, and overall outcomes during induction. And it also prolongs the duration of infliximab with less infliximab discontinuation. The graph below here is from the pivotal study that was used that helped get FDA approval for infliximab in children. As you can see, some of these targets would be now considered below target for exposure during and towards the end of the study. And MIPD software can help achieve more precise targets that are currently acceptable and making the use of infliximab safer and prolong the use of infliximab in these patients. So what's next? We need to continue to think that what is the right dose for the at-risk population, the critically ill, elderly, obese, and pediatric. There is a lack of labeling and guidelines in these patient population. As I mentioned, the FDA Project Optimus has shifted the paradigm for dose selection in oncology drug development, focusing on more dose based on exposure and response analysis and model-informed simulations, allowing for ability to confidently transition the developed models into practical real-world application at the bedside, and continued surveillance for long-term tolerability and model-informed dose modification approaches, which is something that is lacking in the currently approved medications. Pharmacogenomics is another area that will be seen as an expansion for for precision dosing. Pharmacogenomics can have a significant impact in pharmacokinetics, so we need to incorporate that into our MIPD algorithms to further refine and personalize dosing regimen. There are already population PK models that are incorporating this data, such as a drug that is used in transplant for tacrolimus, voriconazole, which is widely used in both oncology patients and as an antifungal, and other critically ill patients, and warfarin, one of the older anticoagulant that is still currently used and still widely used, and we continue to have challenges in monitoring that medication. So in conclusion, precision dosing is a crucial aspect of optimizing therapeutic outcomes and minimizing adverse drug events by tailoring drug regimen into individual patient needs. We need to really integrate precision dosing into drug development and clinical practice, and paving the way for a more individualized patient-centered approach. We need to embrace model-informed precision dosing to support the advanced patient care and use real-world to define dosing for our individualized patient. MIPD is here and it is clinically feasible. The next iteration will be to expand emphasis on integrating disease progression and pharmacodynamics that can result in proactive intervention. And we need to just continue to work in collaboration to utilize real-world data that already exists and continues to be generated every day from our patients to continuously learn and improve patient outcomes. And I thank you for your attention today, and I'll take any questions at this time.
Thank you so much, Sharmeen, for that incredibly fascinating talk. I can see that we have received some questions from the audience already, so we'll get straight onto the Q&A now. So, Sharmeen, your first question is, in your opinion, what is the current status of precision dosing?
So precision dosing, as I've throughout my talk discussed, is increasingly integrated into clinical practice. Is being driven by advances that we're seeing in modeling and machine learning. The current efforts focus on optimizing drug dosing through individualized approaches, particularly for the complex conditions like oncology, infectious disease, and chronic illnesses. And also keeping in mind that our patients are becoming more and more complex, and we need to be able to treat those patients using individualized therapies.
That's a fantastic answer. Thank you so much, Sharmeen. I can see that we have another question from our audience here, which is, what are the top areas for change for precision dosing in 2024 and 2025?
Well, I think, we're already seeing this year with the advent of AI and machine learning, but really in the upcoming years, I do see significant advancements in integration of machine learning into precision dosing. And these technologies will enhance the predictive capabilities of our models and allowing even more personalized dosing recommendation. Additionally, I think the field of pharmacogenomics will continue and is continuing to expand, providing more granular data on how genetic variation includes drug metabolism responses, and further refining those dosing strategies. And lastly, another trend that will directly or indirectly continue to move the needle of precision dosing is the trend towards home-based therapeutic monitoring. So using things like microsampling techniques, which is also expected to grow and make precision dosing more accessible and easier to implement.
Another fantastic answer. Thank you so much, Charmaine. I know that you touched upon this in your presentation slightly through case studies, but are there any other case studies that you can speak to at this time that demonstrate dosing's impact on precision medicine?
Yeah, I think one of an example that I think many would be aware of, as I've discovered, sorry, as I've discussed, many examples throughout my presentation. Another one that comes to mind is PD-1 inhibitors. These are drug medications such as pembrolizumab or nivolumab that are used in oncology patients. These are PD-1 inhibitors. They're essential for treating various cancers, including melanoma and non-small cell lung cancer. Initially, these drugs were dosed based on fixed schedules and fixed doses without much consideration for individual variability in PK and PD. However, the modeling has led to significant changes in dosing strategies. For instance, studies reveal that weight-based dosing, could be replaced with fixed dosing without compromising efficacy or safety. And the shift was supported by POP-PK models that demonstrated similar drug exposures across patients with varying body weights. These models have also showed that fixed dosing approaches simplified administration, reducing dosing errors and minimize treatment costs without affecting therapeutic outcomes.
That's wonderful. Thank you so much for sharing that with us. And we have time for just a few more questions before we close out this session today. And we have quite an interesting question here, which is what are the key thought leaders to impact the last mile of precision dosing?
There's probably too many to name here, but I'll just name a few organizations that I think these thought leaders are all very much a part of. And those are organizations like International Association of Therapeutic Drug Monitoring and Clinical Toxicology, as well as American College of Clinical Pharmacology, and various other organizations that have clinical pharmacologists and pharmacists that are, they're on a mission to make sure that each patient is getting precise dosing, as precise as we can get with the information that we're given, and continuously we're improving on those and evolving as our population is evolving, as our patients are evolving. So I would just recommend keeping an eye on those organizations and the publication and recommendations that come out of those organizations.
That's perfect. Thank you so much. And our final question to close out the session is what results are most likely to be recognized once dosing models are standardized?
I would say that again, better outcomes. I think we can have the ability to see, look at large cohorts of data and continue to improve the predictive power of dosing, is where I'd like to see us move closer to.
That's great. Thank you so much, Charmaine. But unfortunately, that is all that we have time for today. Any questions that we didn't have time to get to will be answered offline as soon as possible. So that's it for this year's online symposium, Advances in Drug Discovery and Development. As mentioned throughout the day, all the talks from today's event will be made available to view on demand for the next 30 days. You can access the talks via the same link you used to access them today, and we will be sure to pass on any further questions that you submit through the system to the relevant speaker. That just leaves me to say a massive thank you once again, Charmaine, and we'll see you again at our next online symposium, Advances in Proteomics and Metabolomics. Thank you and goodbye.